title Biosynthesis of Nanomaterial Infused Lime-Based Concrete and Bricks with Self-Healing Properties.
Just give me a second. Just 1 second.
Very interesting, and you have seen this my academic interest. You are saying some people are in Bangalore and all that. Can you go visit as a student at Kerala? Can we? Okay. Can we put in tickets? So I'll try
A very good morning to one and all. My name is Ritvik. I'm a principal UX architect at Auckland. And I'm also a visiting faculty at several places, including Alliance University. This is the work with my students there as well as my colleague there, Dr. Anirban Chaudhary. And the title of our research is Biosynthesis of Nanomaterial Infused Lime-Based Concrete and Bricks with Self-Healing Properties. This is the overview. We'll look at the introduction, we'll look at what is nanomaterials, we'll look at what is living building materials, what we try to do in this study, some results, and what we are trying to do in the future. So nanomaterials has shown some immense scope in improving properties and creating new kinds of materials, which is called as metamaterials. What it does is, it changes the properties when you're assembling at a nano level. For example, there's the AG9 silver nanocluster. Normal silver is non-magnetic, but the silver nanocluster becomes magnetic. So you have applications now in tagging cells using this nanocluster and extracting cells of interest. We'll look at other examples a little later. In the right conditions, bacteria, viruses, and fungi are known to synthesize nanomaterials. Certain research papers consider bacteria, viruses, and fungi to be nanites or nanobots at that level. Although controlling them becomes a little difficult, we'll not cover that aspect in this paper. And buildings that are made with buildings that are made with limestone are known to last a lifetime. Best example is the pyramids. They've hardly undergone much damage over the years except due to human interventions. Roman concrete that incorporated limestone is known to heal itself. This was proven by researchers at MIT. And there are limestone microbes. So if you have gone to caves, you'll find structures that are known as stalactites and stalagmites. And there are species of bacteria and fungi and other microbes in it that help the growth of this by taking lime from the water that drips in the ceilings, as in calcium hydroxide that's present in the water in these places. And it also takes the carbon dioxide from the air. We find similar structures in urban environments when accidentally sand from these places ends up in construction. So this bacteria ends up over here. In this study, we incorporated these sort of microbes into concrete and made it heal itself. We'll look at that. We also did a scanning electron microscopy, FTIR, that is Fourier transformation, infrared spectroscopy, and X-ray diffraction to confirm the crystallization of calcium. So what are nanomaterials? They're synthesized using specific process at nanoscale. When bacteria, and they have interesting properties, like I said. Example, the AG9 nanocluster, which is magnetic due to the icosahedral geometry of its orbitals when they're aligning with each other. When microbes are used to synthesize these materials, it's called biosynthesis. Even plants under right conditions can do this. And a variety of industries use this: electronics, civil engineering, pharmaceuticals, agriculture. We have some images over here. This one where you can see gold nanowires that has applications in electronics that is synthesized by the Aspergillus fungus. Tobacco mosaic virus synthesizes some nanomaterials, and lactobacillus synthesizes nanomaterials, gold nanorods, by a variety of researchers. So these people believe that nanobots, which was a figment of science fiction, can be made into reality with the help of these microbes. Because if you look at it, if you look at science fiction, these nanobots can help buildings heal, help materials heal, can assemble things at a nanoscale. We've got bacteria, viruses, and fungi doing it. You need to genetically modify them a little bit if you want to control their movements, or you can use something like an 89 nanocluster which has a peptide attached to it. Now, if you imagine that peptide is an antibody, it can attach itself to a virus or attach itself to a bacteria, and then you can control the movements using magnets. We'll not look at that in this research because that's not our aim. What are living building materials? So when you incorporate such sort of microbes into building materials, they're called as living building materials. So there's bioconcrete that absorbs CO2, reduces greenhouse gas emissions. We have that thing as well, but although this one's a little different from what we did. The Chettinad plaster, this was something done by the Chola dynasty. If you've seen Ponniyin Selvan movie, it's the guys from that. They incorporated both limestone bacteria and curd, lactobacillus from curd. Now back then there was no microscopy or anything, it was just based on intuition. The beauty of this is, the curd, they mix some sort of jaggery, plant material, other things in it, so that became the food for the curd bacteria. It used to generate CO2. The limestone bacteria's food is carbon dioxide, along with lime and other things. So this plaster became strong over time. And that is why you see these temples that are built by them are still extremely strong, whereas the Ejipura flyover keeps cracking. Mycelium and rice husk has been known to make bricks that are durable and fireproof. This is by researchers in Australia. So what we did, we collected some limestone growths from cellars. We thought, let's see if we can make self-healing bricks, see what happens. And we wanted to confirm whether it is really crystallizing calcium carbonate. So we did a variety of things: X-ray diffraction, Fourier transformation, infrared spectroscopy, and of course, scanning electron microscopy. So we went around. These are from some cellars in Bangalore where we found these limestone deposits, limestone growths. We put on some gloves, we used a hammer and chisel, carefully collected them, and we grinded them carefully. And we created bricks with unique compositions. So there was one with pure concrete that served as sort of a control one, and pure limestone, the second control one. The third control was limestone plus concrete. Then we created a brick with concrete plus these growth material, limestone plus these growth material, then concrete plus limestone plus growth. We wanted to see which brick would heal the best, which mixture. Of course, the first three wouldn't heal much because, there's no limestone bacteria. Then what we did is we created fractures in them. These are the various bricks that you're seeing. Is there a pointer here? Okay. So these are the various bricks that are there. It's okay. Then what we did was we placed these bricks in a lime water drip. So you can see this thing over here. This is the same thing that's used in the drip irrigation. So water drops, a few drops for an hour. In 1 hour, you'll find like 5 to 6 drops of water. And that had calcium hydroxide infused in it. And we put this setup in a room temperature of around 25 to 28 degrees centigrade. And people used to come and go in this room and the room around it. And we used a pure cement and pure limestone bricks as the control group. And to confirm whether there was mineralizing, we used, as I said, and these were the various instruments. These were all present from the Sophisticated Testing and Instrumentation Center, the Alliance University. We thank them for helping us with this research and letting us use these equipments. What happened after a few days was, you can see the before and after effects. So you can see all these cracks, that's practically healed over here. I've placed some bricks, you can take a look at that. You can see the difference between the third one and the corresponding one below it. You can see such a huge fracture has completely become small. And similarly the one over here, all those small fractures that are there, practically gone. And the brick that worked the best was something that had incorporated both cement and limestone and the growth material. FTIR spectroscopy confirmed the presence of calcium carbonate as the frequencies for the same showed up in the spectrometer, which is around 700 to 900 by centimeter, and we had specific peaks are sharp at 713 and 865 centimeter. So it indicates the presence of amorphous calcium carbonate at these 1419, 1420, 1470, and another one at 1490. X-ray diffraction also confirmed the presence of calcium carbonate with around 84.92% accuracy. Then we had the SEM images. Now this was the beauty of it. Mineralization of various, not just calcite. We were hoping that, we'll just find calcite, but we found calcite, valerite, and aragonite. So it's not just calcite crystals, it was crystallizing other forms of calcium carbonate at a nanoscale. That was the beauty of this. Future work, we would like to experiment on the growth of this bacteria on Martian regolith stimulant. So that's Martian soil, simulated Martian soil. It is sold by this company called Space, which is endorsed by NASA. So if we are building colonies in Mars in future, we'll have to ensure that any holes or any fractures that are found can heal itself. The idea is to use limestone microbes and see if colonies can be built on Mars. Questions?
Thank you, Professor Ritik. You are from which university?
So I am a principal UX architect at Optum. That's my full-time job.
Oh, okay.
Yeah.
Okay.
And Optum's a pharma company. Cephalosporium is also something of interest to us because it's used in
So
pharmaceuticals.
you are in collaborating with Alliance School of
I'm a part-time faculty at Alliance, Narsimon University, and Presidency University.
Any queries or sessions from delegate side?
Oh no, that's the next one. Because limestone is very fragile. If you don't mix it properly, it can cause issues. Concrete is strong. But again, if you're not mixing the right, too much sand will make it brittle. Too little sand will also, make it brittle. It's about getting the right composition. So that is the next part. We want to see, okay, this can heal itself. It can absorb some CO2. But is it strong? And how do we improve the strength? We might want to take some inspiration from the guys in Australia where they added rice husk, other sort of materials into this and see how we mix and match. So that's the next step. Apart from the Martian simulant.
Any idea what type of instrument you do also?
That I'll have to determine, drop tests, that sort of stuff, basic ones, but we want to even test it using those hydraulic presses and see how much force it can take.
Any other queries?
You were talking about that you are using microbes as a nanomaterials
Yeah, to generate nanomaterials.
to generate nanomaterials.
Yes.
And it is, you are saying that it is you are able to, you can control the amount or the
Yeah.
conditions.
So, I didn't want to cover that in this research, but if you apply an electric field, not a very high electric field, a certain voltage to bacteria, you can control their multiplication rate. You can increase it or decrease it. So that is one way of controlling. The second way is, what is an antibody?
Yeah.
So an antibody is sort of a marker for a white blood cell to come and eat a bacteria. And there are specific antibodies for specific types of bacteria. So let's take, or they're peptides basically. So this peptide can be attached to the shell, the outer shell of that bacteria, right? It gets attached. Now let's assume that you're attaching another, nanomaterial at the end of it. So AG9 nanocluster cannot exist on its own. It requires a peptide chain to exist along with it. So there's a chain of these carbon compounds का एक chain होता है, and around that you have an AG9 nanocluster that is formed. So imagine this antibody as that peptide, and this AG9 is on it, and that goes and attaches to that bacteria. Now I said AG9 is magnetic. So if I put a magnetic field, I can control the movement of this. We don't want to do that. I mean, you can do that in a very nice small petri dish. And for example, let's assume this is a copper-eating bacteria, right? There are microbes that eat copper. So you attach these antibodies to this bacteria, you leave it with some copper compound, it will eat the copper. Then you put a magnetic field, you'll be able to pull the bacteria towards one side. You add a solution to kill it, you'll get pure copper in your hand. These are experiments that have already been done at a lab level. So this is the next step, but we don't want to go there yet. We want to try out things and then go to the next complex level of trying to control the movement and control its multiplication rate.
Yes. Okay, when you see, I have one.
Yes.
Go to the previous slides. Where you made certain mixers, right? Yeah. Okay. So you consider this pure concrete, pure limestone, and these are six components you consider, right? Okay. For first two is okay. For next four, do you consider any volumetric ratios for this mixer?
I'm not able to recollect, but we followed the specifications that was there in the packaging.
Okay. Because the point is that if we change the volumetric ratios, because you are getting optimal values for fourth one, I believe, right? Sixth one, right? Okay. So if you change these volumetric ratios, there may be some, higher standard of solar panels.
So I think the growth was around 50 grams and concrete was around this one was like I think 100, 150. The total weight was around 250 grams. You can check this here.
Okay. No issues. Okay. Great.
But your point is right. Too much limestone or too much concrete will alter how it repairs itself or it will alter the strength as well. That is something that we want to check in the next phase.
Yeah. Great. Thank you, sir. Now I'm going to the next paper, ID, Dr. Nitin Gaur, developing an integrated framework for physical and digital applications with the use of AI and advanced technologies for effective mass communication.
So I'm going to leave this here. If others want to
Yeah,
take a look,
yeah.
we can.
Please use here.
Sure. That's it. No. You can note down the number. Often doesn't hand out visiting cards. 6361 28 7376. Yeah. That's what Ramadas. Yeah. OPT. EU. From Kerala, born and brought up in Hyderabad, studied in Hyderabad.
Good morning. I'm Professor Nitin Gaur from Symbassi Institute of Design. Our subject